Hello boys and girls, welcome to another flipped video. Tonight we are looking at a dot point which you might find some similarities to other dot points. It's about waste and waste removal and what this means for metabolic activity, so let's have a look. Unpacking the dot point, our verb is explain, so we have to provide a cause. So we're looking at if we don't remove the wastes, what impact does this have on metabolic activity? So that's going to be our response to explain why the removal of waste is essential for continued metabolic activity. Alright, let's quickly look at what are the waste products of the body. So the first one is sometimes in a chain of reactions, one of the products that is produced from an enzyme regulated reaction is not needed by the body. It's kind of like the chippings. If you imagine like someone sculpting a molecule, it might be one of like the shavings on it type thing, so it's kind of discarded by the body. Another one is nitrogenous waste, and in the human body this is urea and other smaller trace things like ammonium and creatine. We're also looking here at carbon dioxide and water, and then excess salts. So a bit of a conversation about it today. You would have done a fair amount of carbon dioxide already, so we should I think we'll nail this one pretty well. Alright, so I wanted to talk about this first one that we talked about, which with the unnecessary products of a reaction. So let's say that this is this little substrate is our clay, and it is being enzyme 1. It turns it into our product 1, and this is also called an intermediate substrate. And it gets molded and molded and molded, and eventually it turns into these two products here. Now, what the body needed was this orange and blue one here, and its end product, that is the square one, is kind of unnecessary. And I want to show you what this can do to an enzyme. It's called inhibition, and what can happen is they can bond with the enzyme and change the active site. And when you change that active site, you guys know that you cannot bond with the substrate, and so you break this cycle, this idea of inhibition. If you have too much of something at the end that is not being removed, it can come back and inhibit the original process. So your body's got to have some ways of removing these things. Alright, so we're looking at other reasons to remove them, and it's this toxicity. And when we think of toxic, we have these images appear in our heads, and it's really a little bit too extreme. What we're looking at here is a conversation around nitrogenous wastes. These are toxic. Toxic is basically a build-up of something. Anything can be toxic. Toxic is a term that's based on dosage. So in large enough doses, oxygen is toxic. In large enough doses, water is toxic, and I don't mean drowning. Like you can have too much water in the body. So when nitrogenous wastes are in the body, they change the pH, and so they fall. The conditions fall outside of the optimal range, and we know that when that happens, enzyme function and activity decreases. They can also actually interfere with membrane transport, and this is pretty important when we realize that we have those sodium and potassium pumps in our membrane, and that they can move substances in and out. And so if you interfere with that, you kind of not only interfere with the removal of nitrogenous waste, but you're also causing like a chain reaction with other things. So it's a pretty important substance to remove, and yeah, very good. All right. So now we're looking at carbon dioxide. You're very familiar with this one, but I thought I'd go into a little bit extra detail with something which I think will help explain it a little bit more in terms of disease. So we know that we lower the pH with this when you have too much carbon dioxide. But what this actually in the long term builds up is it's a condition. So in the extreme buildup of carbon dioxide in the body, you get acidosis, which is the lowering of the blood below 7.35 pH. And it can cause two things. You can have a respiratory acidosis, and this is clearly about blood pH, and then you can have metabolic acidosis, and this is where it affects the kidneys. Now, both these organs are organs of waste removal. We are removing carbon dioxide and water through the lungs, and we are removing water, excess salts, nitrogenous wastes, a whole bunch of stuff from with our kidneys. And so if you fail to have these working properly, you start having symptoms of things like drowsiness or fatigue. You've got, with the metabolic acidosis, you can be jaundice, which is a yellowing of the skin. And the jaundice is a really concerning. It's visually concerning, but it's a pretty significant poisoning of the body with materials that should have been removed. So I think in terms of kind of your understanding for the course, I mean the key thing is obviously that it lowers the pH, and that can have the flow-on effects for the enzyme function. But our body has systems in place to kind of handle that short-term impact. It's when we have systems in place to fit within to operate within our narrow range of tolerance. But when that tolerance is broken or tested, we can have this long-term build-up and we get acidosis. Alright, so let's have a look at our excess salts. You might have seen a diagram like this from year 11. This is basically what happens if you have salt outside of a cell and salt inside of a cell, like excess salt. What happens to the water movement? Because we know that water wants to move to create a balance, an osmoregulated balance. So we've got in a red blood cell, if you have excess salts inside the cell, you are going to have water inside that red blood cell. The red blood cell is a very weak cell. It's a very flimsy balloon, I think, and they can burst. This is called lysising or lysis, and that's not good. In like maybe a cheek cell, you get some more swelling here. Now the swelling may not have a huge impact, but it can. And you'll see here that we have a neuron. So this is like one of our nerve cells that is transmitting messages. And if you're having an impact on the amount of water going into a cell, then you're going to have an impact on substrate concentration. And we know that when we alter substrate concentration, we are going to have changes in enzyme function. It's really, we can't kind of nail this enzyme function more. It's a pretty important concept. When you have too much salt outside of a cell, you have the water leaving the cell and cells shriveling. So if we have here, it's all about substrate concentration and actually having a solute that allows you to have an interaction with molecules. So if you have too much water leaving the cell, they shrivel. This is also not good for general metabolic activity. Okay. Alright, so in a bit of a summary, I thought I'd just kind of like make it relatively simple for you guys. We're looking for big picture understanding here. The understanding is that metabolism produces waste in a variety of different forms. Every single waste product can, in some way, shape or form, change the effectiveness of metabolism. Namely, that they affect enzyme function in a negative way, changing the shape, reducing their ability to bond with substrates, lots of different ways. And metabolism is the term given to chemical reactions that sustain life, and if you're not metabolizing efficiently, you will cease to live. Looking at some of the details though that we want you to kind of like be able to throw into your responses, depending on how you choose to form a response, you want to talk about things like changing shape, reducing ability to bind to substrates. Nitrogenous waste such as urea can change both the pH and the ability of membranes to transport materials in and out of cells. CO2 lowers pH, and excess salts can affect osmotic pressure. If you can get the big picture first and then kind of like find a way to weave those details into some sort of response, then you're going to have, I think, a pretty good time with this dot point. Okay, have fun, and talk to you in class.